Good morning to all the students, and today we are going to start with sexual reproduction of flowering plants introduction. What are you going to learn in this chapter? I will be telling you first, and then we will move on to the concepts that we are dealing today. Sexual reproduction of flowering plants, if you have to fragment this into three parts, in this chapter you will be learning about pre-fertilization events, fertilization events, and then post-fertilization events. First, pre-fertilization events, what you will be learning here. Have male reproductive structure and female reproductive structure, how male gametophytes and female gametophytes are formed, what will be the male gamete and female gamete, the complete formation and the process of that you will be learning in pre-fertilization events. What you will be learning here first will be microsporogenesis, formation of a male gamete, and second is megasporogenesis, formation of a female gamete. Complete processes you will be learning. And before moving on to the fertilization events, here you will be learning on the types of pollination or the modes of pollination, and pollen pistil interaction, post which you learn on double fertilization events. About double fertilization events you have already learnt in your 10th class, right? What is a double fertilization? How is it happening in angiosperms? You have already learnt in 10th. But here along with fertilization you will also learn on pre-fertilization events, post-fertilization events, and special types of reproduction like apomixis and polyembryony. In fertilization events you will be learning on double fertilization. Post-fertilization events, if you look into seed formation, seed is formed from the ovule, fruit formation, prior to these two events, two more events happen, that is embryo and endosperm development. Got it right? So, these are the major events that happen in sexual reproduction of flowering plants. Along with this, you will be learning on some special phenomena, that is apomixis and polyembryony. Fine, right? So, be it from pre-fertilization events or fertilization events or post-fertilization events, or even from the special phenomena like apomixis and polyembryony, from any of the concepts, we cannot weigh like this concept is more important and this concept is less important in this chapter. All concepts of this chapter weigh equal weightage, and what is the probability of questionnaire that you will get from this chapter in the final exam will be around 3 questions, 3 to 4 questions for sure. It will be around 3 to 4 questions. And in today's session what we are dealing is the basics of sexual reproduction of flowering plants, like how a flower is formed, what is a male reproductive structure, female reproductive structure, what are the components of male and female reproductive structure, how will this happen, and then we will learn a story at the end on how the pollination happens, double fertilization in gist, and how an ovule is formed, ovary is formed, from that a seed is formed, a fruit is formed, everything we will learn it in the form of a story at the end. So, you will learn it in a very easy way, focus till the end. First, if you consider flower, we know flower is the reproductive organ of the angiosperms. Flower is the reproductive organ of angiosperms. Flower is the reproductive organ of the angiosperms. Right. And this flower can be unisexual or bisexual. Unisexual or bisexual. Unisexual, it can either have male reproductive structure or female reproductive structure, any of that will be having in the flower. But in a bisexual flower, you will see both male and female reproductive structures. Male reproductive structure, male reproductive structure and female reproductive structure. Male reproductive structure is addressed as andrecium. The unit of andrecium can be a single stamen or multiple stamens. Okay? So, the unit of andrecium is stamen. Female reproductive structure is addressed as gynoecium, which is also known as pistil. This pistil, what is the unit of gynoecium or pistil, is carpel. Means a pistil or gynoecium can be monocarpellary, multicarpellary. Here if you observe the components of stamen, it has one long filament and anther lobe at the top. Okay? Anther lobe and filament. Fine, right? It has an anther lobe and a filament. Anther lobe is dithecous. It has two theca. We will clearly learn on the structure of anther today, and it has long filament. If you look in the diagram, here you can see this is the long filament and this is the anther, dithecous anther. This is addressed as, at this region we will call it as the proximal end of the filament. Proximal end of the filament is adjoined to the thalamus and the distal end of the filament is anther. Proximal end of the filament is thalamus, distal end of the filament is connected to anther bilobe. Proximal end to the thalamus, distal end is connected to the anther bilobe. This is about stamen. Now, if you consider a single carpel, you observe here there are three components of carpel. Fine. This is known as stigma and this is style, stigma, style and ovary. Inside ovary you have placenta. Inside ovary what is present? Placenta. Like how in a mother's womb placenta is present, same inside the ovary. This is the basal bulged portion. Inside the ovary you will be having placenta. Inside placenta ovules are arranged like this. Ovules are arranged. Inside placenta ovules are arranged, right. So, the basal bulged portion is known as ovary and inside the ovary fluid filled space is known as placenta and placenta will have ovules, right. So, here can you see once again, let me recall you. A flower is a reproductive organ of an angiosperm. It can be unisexual or bisexual. Unisexual can either have male or female reproductive structure, any of them but not both. Bisexual flower will have male reproductive structure and female reproductive structure, both of them. Male reproductive structure known as andrecium, units of andrecium is known as stamen, and the components of stamen, if you see, filament and anther lobe. Proximal end of the filament is connected to thalamus, distal end is attached to anther lobe. Female reproductive structure is known as gynoecium or pistil. Pistil can be monocarpellary or multicarpellary. A single carpal unit, if you see, we have stigma, style and ovary. Ovary has fluid filled space which is known as placenta. In the locules of placenta, you will have ovules. The arrangement of ovules inside the ovary is addressed in the concept of placentation that we have learnt in morphology. We will be learning in morphology. Got it right? Till here it is clear. Now, look at the essential and non-essential worlds of a flower. Two reproductive structures we have discussed are essential worlds of a flower. Right? Just a second. Flower, essential and non-essential worlds. It has 4 worlds. 2 essential worlds and 2 non-essential worlds. Essential worlds include andrecium and gynoecium. Non-essential worlds include calyx. Units of calyx are sepals. And second is corolla. Units of corolla are petals. Fine, right? Units of calyx are sepals, units of corolla are petals. This is the complete intro on a flower. And now we will see how a flower is formed. How a flower is formed inside a plant. On a plant, how a flower is formed. The decision of a formation of flower is far before the flower formation. Even before the flower formation, the decision of making a flower is decided by the plant. How will this happen? Consider this as a rough diagram of a plant where we have buds, axillary buds and apical buds, right? Apical buds we have and axillary buds we have. So, these auxiliary buds or apical buds can turn into a shoot branch, can turn into a flower, it can turn into a leaf, it can turn into anything based on the plant requirement. If the shoot apical meristem has to turn on to floral meristem. Floral meristem, a hypothetical hormone or a chemical substance which is named as florigen will be acting on it. What will be acting? Florigen will be acting on it, which is addressed as a hypothetical hormone. Shoot apical meristem will be modifying or turning into a floral meristem. The modification is by the usage of a chemical substance or a hypothetical hormone, that is florigen. What is the change that you observe? Internodal condensation. Okay, internodal condensation. Fine, right? This floral meristem via internodal condensation, this turns out to be a floral primordia. Floral primordia or primordium. As I addressed this as floral meristem, this turns out to be a floral primordium. For example, this axillary buds are moving into some lateral branches from the same auxiliary meristems. The internodal lengths will be long, whereas the modified floral meristem, internodal length will be very less. So, via internodal condensation, this is forming out to be a floral primordia. From floral primordium, this forms out to be a floral bud, and then floral bud matures to a flower. Floral bud matures to a flower. Got it? So, here shoot apical meristem with the action of the hypothetical hormone florigen turns to a floral meristem. Then via internodal condensation, reduction in the length of the internodes, this turns out to be floral primordium, then it matures to a floral bud, and lastly a flower is formed. Clear, right? And what are the components of flower? Essential, non-essential whorls, unisexual, bisexual flowers we have exercised. Today let us target microsporogenesis in detail. Okay, microsporogenesis. Here you observe carefully, this is the stalk or filament at the basal portion, this is adjoined to a thalamus, and at the apical portion or at the distal portion you have bilobed anther. This bilobed anther diagram, if you have to take, this will be the diagram. Right. So, here this is the bilobed anther, this is addressed as lobe 1 and lobe 2. Each lobe will be having 2 theca, this is theca 1 and theca 2, theca 1, theca 2. That is the reason this is known as dithecous bilobed anther, or on whole you can also call it as tetrathecous condition. Okay, it has 2 theca, and inside each theca there is a sporogenous tissue at the center. At the center there is a sporogenous tissue. Fine. So, this is the bilobed anther, each lobe will be having 2 theca, and at the center of each theca you have a sporogenous tissue. And here if you observe the layers of anther is covered by 4 wall layers. Outermost wall layer is known as epidermis. Epidermis is of parenchymatous tissue. It is of single layered and parenchymatous. Then after epidermis, next layer is endothecium. Endothecium is also single layered, it is made up of alpha cellulosic fibrils. It is single layered and made up of alpha cellulosic fibrils. And then furthermore it has multi-layered condition which is known as middle layers. Next, multiple layers, those are middle layers. Got it? Outermost layer is epidermis, followed by endothecium which has alpha cellulosic fibrils, then 2 to 3 layered of middle layers. All these are binucleate condition. Okay, sorry, all these layers are uninucleate condition which are in diploid state. But then the innermost layer is known as tapetum, which is of multinucleated polyploid condition. Multinucleated polyploid condition. This is multinucleated and polyploid because this provides nourishment to the sporogenous tissue which is present at the center. Tapetum will provide the nourishment to the sporogenous tissue which is present at the center. Whereas the other 3 layers, that is outermost epidermis, next endothelium and middle layers, together they form protective in function. Together they are a protective function and they help in the dehiscence of pollen grains. So now you have clearly understood the story of anther. Anther is bilobed dithecous. Each theca will have a sporogenous tissue which is surrounded by 4 wall layers, outermost epidermis, then endothelium, middle layers and tapetum. These 3 layers together are protective in function. Innermost wall layer is tapetum, which is multinucleated and polyploid. This will provide nourishment to the sporogenous tissue which is present at the center. Okay. Now, what is that happening inside the sporogenous tissue? Yeah, inside the sporogenous tissue. The cells which are present inside the sporogenous tissue are known as spore mother cells. This is a sporogenous tissue. The cells which are present inside the sporogenous tissue are known as spore mother cells or pollen mother cells. They are known as spore mother cells or pollen mother cells. This spore mother cells inside which, if you consider this as a microsporocyte, this is also known as microsporocyte. Fine. Spore mother cells, pollen mother cells and microsporocyte. A single microsporocyte, if I have to take a single microsporocyte, it undergoes meiosis 1 and meiosis 2. That means reductional division happens. Microsporocyte which is diploid after reductional division, this forms 4 haploid microspores. 4 haploid microspores. These 4 haploid microspores are associated in the form of a tetrad condition. These 4 haploid microspores are associated in a tetrad condition. Fine, right? This tetrad condition, if you see here, tetrad condition, these are what? Pollen mother cells, and outermost layer of this is what? Tapetum, which is multinucleated or polyploid condition. Here you see this tetrad formation inside the microsporocyte, but then these microspores are not dissociated yet, they are in the form of a tetrad. To dissociate the wall, this wall is known as callose wall layer. To dissociate the callose wall layer, an enzyme action is required, that is callase. This callase is also sent or this is secreted from tapetum. Okay, this callase enzyme is also secreted from tapetum, which will degrade the callose wall layer, and the microspores are liberated into the sporogenous tissue. Now, these microspores need to be matured to a pollen grain. These microspores need to be matured into pollen grain. Microspores matures to pollen grains. These pollen grains, which are in the matured form, are of binucleated condition, but here we have seen 4 sporogenous tissue initially in 2 lobes. Each lobe has 2 sporogenous tissues, but here if you can observe from the sporogenous tissue, this is known as line of dehiscence. In due course of evolution from microspores to pollen grains, this will turn into, in due course of evolution from microspores to pollen grains, this entire thing will be turned to a pollen sac. This will be turned to a pollen sac. Fine, right? And each sac will be liberating out the pollen grains. Got it? This is how the male gametophyte, that is pollen grains, are released into the environment, okay, from the anther. This is the complete formation of microsporogenesis. What does it mean? Formation of microspores is microsporogenesis, and microspores will evolve into pollen grains by taking up the nutrients from tapetum, and in due course of evolution the tetrasporangiate condition will be converted to 2 pollen sacs. From each pollen sac, pollen grains will be liberated. So, in today's session, what are the key takeaways? Initially, we have clearly learnt on the fundamental insights of sexual reproduction of flowering plants and the complete information on a flower formation, components of a flower, unisexual, bisexual, and then we have also learnt on essential, non-essential words, after which we have focused on to the anther structure, and then we have learnt on microsporogenesis. In the next session, we will move on to megasporogenesis. Thank you.